Hello and welcome. My name is Mitch Sanders. I'm co-chairing this really fun podcast for the Wound Healing Society where we feature awardees with Jordan Yaron. Jordan, welcome.
Good to see you, Mitch.
And today we have a very special guest. He's Craig Tipton. He is the Director of Biostats at RTL Genomics, and he got his PhD at Texas Tech and a BS at Angelo State University, yes? Oh, in biology, right?
That's right. The
Craig,
other ASU.
yeah, I was like, that's not the same ASU that, yeah, I was thinking that too. So Craig, in order to get started, why don't you tell us a little bit about your background and how you got involved in kind of wound pathogen microbiome work to start?
Yeah. It's funny because really, if you'd asked me, several years ago if I thought this is where I'd be, it would be a hard no because so initially going into my degree in biology at ASU, I was actually pursuing pharmacy at the time, and I was working in a pharmacy, and I had a pharmacist pull me to the side and say, go do something else, and he's had this really dead look in his eyes, and I was like, you know what? Maybe I'll go for something more sure. And so somehow that led me into grad school. But through all of that time, I was actually, my base background in undergrad was more in just molecular biology and actually more like wildlife ecology. My undergrad research was all in bats, doing that kind of a thing. When I graduated, I landed a spot at Texas Tech in the lab of Dr. Caleb Phillips doing, initially I interviewed on things like looking for viruses in bats, and we started to feel like a certain project was not going to work out. And I remember having this conversation where someone said, yeah, we have some of these weird coronaviruses in bats that we're picking up, but I don't think anyone's ever going to care about these weird coronaviruses. It was back in like 2017, and
and the COVID hit.
Yep. So it's funny the way things work out. But around that time, I was kind of shown we have this other thing that we're working on, though, with this local wound physician. His name was Randy Wolcott, who a lot of people in the WHS are probably very familiar with. But Randy had just been collecting so much data over the years, and in a lot of ways was kind of pioneering this approach that he was developing of he wanted to sequence all of the different microbes and things that were showing up in wounds, and he had the I think kind of against the grain idea at the time to sequence the wounds, get an idea of what bacteria there, profile some of the resistance genes, and actually target. I think he started with systemic and then moved to topical antibiotics based on what we were finding there, and it seemed like he was having a lot of great success with that approach. But he had all this data he'd been collecting over years, and so kind of approached us about kind of collaborating because our lab, or the advisor that I worked with, Caleb Phillips, he was kind of a guru in all things metagenomics. And so I was there learning all of the different steps around identifying, sequencing, and then processing all of that data. But I still had that background of ecology in the back of my mind. So while we're looking at the consequences of these bacteria that are there, I'm also always wondering about questions related to, well, why are they there? And why are there different bacteria there? And that was always interesting to me. And that led to one of our first or earlier kind of big papers in this space, which was comparing patient genomic markers against those profiles. And we found certain signals associating, explaining that little differences in the genetics of our patients could explain the likelihood of seeing, say, more Staph epi versus Pseudomonas in a wound. So that was kind of one angle. But then taking it a little bit further as we were going, there had always been this kind of small debate in that process of, at the time, you could look back and most of the wound microbiome work was really happening in three places. It was like Lubbock, Texas. I want to say somewhere in Florida, and then somewhere in the oh gosh, I'm forgetting a big one right now. I want to say it was in the Madison, Wisconsin area.
Yeah,
At least in the
Wisconsin's big.
United States, those were some of the big ones. and you would see different bugs show up in the different areas. And it led to some conflicting opinions of, well, like, you're only seeing Pseudomonas so much in Lubbock. Like maybe that's just a bystander, maybe it's not real. It's a lot of different conflicting opinions. But this led me to have these questions of, could there be something that's causing that? Are there legitimate geographical differences in that? So that's kind of what led me into developing that particular project. But what set the ground for having access to, thousands of samples the whole time while I was attending grad school, and for the several years past, I've kind of had one foot in academia and one foot in more of the industry side the whole time that I was going through this process. So during grad school, I started working part-time for RTL Genomics. That's the company that I still work for now, on more the data analysis and biostat side. So I got to work with a couple of great people, learning to get more heavy into the biostats and the data analysis and the bioinformatics, while also still attending tech and working on these different things. That led me to a point where I'm going to intro another company who eventually acquired RTL Genomics in this cycle, Microgen DX. Microgen was a company that spun off from Randy Wolcott's clinic originally. So he had that idea of sequencing wounds. Someone else came along and thought, this is really great. I want to expand this to a much broader scale. And so that's kind of the very brief story of how Microgen DX spun off from a single clinic into being kind of a national reference lab. But Microgen DX similarly was just collecting all kinds of data, binning it by different types. And so I was able to approach the CEO and essentially ask for access to a lot of the wound data, because following this question and this idea of, are there different geographical patterns in the different bacteria that we see, and can we associate them to anything? They had been receiving samples from most every state in the United States. So that's the story of how we got these 10,000 or so samples that was used in the abstract that was submitted to the WHS. And then because I felt like that just wasn't enough data and wasn't enough work to do, I had the crazy idea of, what, maybe if there is a geographical pattern, maybe there's something driving it. I wonder if we can somehow link all of the local weather data from all of these different clinics to the different microbial patterns that we're seeing, which kicked off about a year's long process for me trying to figure out how to get and work with that data. It was almost just as challenging as any of the bioinformatics of processing all that sequencing data. Led me to work with another faculty member at Texas Tech in the atmospheric science division, who I spent 7 or so months trying to figure out how to get this data, and I sent this professor a cold email thinking I wouldn't hear anything from her, and instead she wrote back and said, "Oh, I have all this already."
That's the best data ready to go.
Yes, it was amazing, and she was a huge blessing on that. But so that's kind of the story of how I assembled this large hodgepodge data set, and then I'll come back to that study in a minute, but just to finish up some of the background. So going through that process, I finished my PhD and then went on to work full time for RTL Genomics, still doing the consulting. So that job is a lot of fun in itself, just out of these kind of the skill set that I've developed over the years doing this is something that is, I think, sorely needed. We've entered this age of data where there's more data than people that know what to do with it all. And so that analysis is a really in need skill. So we get to work with a lot of different collaborators across the country. And then the part where RTL was acquired by Microgen, I transitioned really well, since I'd already been working with Microgen data for a few years at this point, into kind of helping to design application studies to help demonstrate the utility of what we're doing and the utility of kind of next-gen sequencing, and actually helping to improve patient outcomes. So that is a really kind of exciting thing in its own for me. But that's where I've been. That's where I'm going.
So let's jump back into the paper and understand the data a little bit. And you said this is coming out in Nature and it's in review now?
Yeah, so it's in review right now at Nature Communications. I'm actually in the process of getting that resubmitted, hopefully in the next week or two. They asked for a couple of tweaks, but when you're dealing with 10,000 samples, tweaking a couple of things can be a little slow sometimes. So that's where that's at right now.
And can you talk about the kind of major developments around the 6 different microbiomes that you saw and the implications of the data as well?
Yeah, so jumping into that, definitely kind of the first major goal with this data set was we just wanted to understand, what does that core microbiome look like in the wound microbiome nationally? Can we identify particular types? And so to go about that process, I first started with this clustering method. It's called the Dirichlet multinomial clustering process, which just kind of takes all of these different community profiles and tries to shuffle them into a small number of distinct community state types. This method, I think, was first used in helping to distinguish gut microbiomes that did or did not associate with causing cancer, had a lot of success and utility there. And then I wanted to apply it here. And we saw a few patterns and a few really interesting things that stick out. For one, one of the really dominant community types was actually just pretty simply Staph aureus. Like if you separate all of these microbiomes into 6 types, one of them is just mono Staph aureus, which I think for people who've been studying wounds, that is not a huge surprise.
Right. Yeah.
But when you break into the other ones, some of the really interesting patterns that occur. So we are able to kind of compare this data also with measurements of the wound microbial burden through quantitative PCR. So for all of these communities, we had some estimate of the number of bacterial cells there. And 4 of the 6 of those were well above kind of this, it's not perfect, but there's kind of this magic rule of thumb of if the community is above, say, 10 to the 5th grams of bacteria per or
10 to the 5th CFU per gram.
Yeah, thank you. It's kind of a magic cutoff for this is likely a load that can cause infection, and this is a burden that is maybe worth considering treating with antibiotics. There's some controversy around that aspect of it, but that's kind of the cutoff used for infectious or not infectious.
Yeah, the old notion of critical colonization goes back to like Phil Bowler. It goes back like 2000. And that was before we thought about biofilms. And we've been studying kind of polymicrobial wound biofilms for, I don't know, 20, 30 years now. And we understand that, you have at least, the average is what, 2.8 organisms per wound minimum. So you probably have at least 3 major species in the wound. That's not counting all the anaerobes and other things that are in there too. Whereas you're counting everything.
Yes, and I'm glad you bring up the anaerobes and the polymicrobial nature, because I mentioned the one kind of just Staph aureus dominant community type, but what we saw in all the others was that you had different mixtures that could lead to kind of more of the polymicrobial aspect and different assemblages that led to high bioburden. And one of the things that stuck out to me was there was 2 different community types that were kind of characterized by this increase in the abundance of anaerobes. And anaerobes really seem to be the key to driving a high diversity and high bioburden wound. And of those 2 types, they seem to be further characterized or dichotomized into those with and without staph. So even where staph isn't the key player, it's still present in a lot of these other types. And so there was one type that was, you can kind of think of it as Staph aureus plus anaerobes and all the diversity, versus one that was more strictly about just a high abundance and a high load of anaerobes. And then there was one of the other high load communities was, in my view, it was more of just the mixed bag type of, it could be anything else that.
How often did you see polymicrobial events with like Candida albicans and other bacteria in there?
So the little bit that I did kind of looking at Candida was somewhat limited, but what I did see was that there were certain community state types that were up to 2 or 3 times more likely to have Candida albicans in it as well. I'm forgetting which the exact types were right now, but there was that pattern of some of these types were associated more with Candida albicans. So it's definitely playing a role.
So the other thing that's interesting is I've seen over the last 20 years a shift in the hospitals of what organisms are showing up. And part of it is the antimicrobial resistance is kind of playing an important role. The other role potentially is the synergistic effects of these microbes and how they can kind of coexist in a deep tissue biofilm, right?
Yeah.
So
Yeah.
where do you think the research will bring you next? What are your next big questions that you'd like to answer?
Yeah, so I mean, just on this, not even getting into any of the like weather analysis that we did. The next big thing I would like to do just with these community state types is, so with this particular data set, we're working only with what our lab really had access to in terms of, we know that there's a patient, there's a profile. We don't know anything about um, that patient's wound, their other comorbidities, their trajectory. What I would really like to do is kind of replicate this in, um, at least one of the intentions that we have right now is to limit this to one single center analysis where we have access to a good number of patients over a couple of years, where we can kind of replicate these community state types, see how often they're appearing, kind of look at what transition between those types looks like. And if, a hypothesis that I have right now is that if you see a transition from one of these high bioburden, what I think of as like the really bad types, to one of the low bioburden types, my hypothesis is that you would also couple that with improved rates of healing and better outcomes over time.
But
Um,
could it also be the amount of proteins that they're secreting? So strep pyogenes, by example, you can have less than 10 to the fourth in a wound, but it's dumping out so much protease, it's really bad. And so it also depends on the virulence factors that these microbes are putting out and how they kind of work together.
Absolutely. That is one thing that it's a hard aspect to capture just in this data set, because the 16S sequencing that we're using, it's only able to
ID
really
them all.
characterize.
Yeah.
Yeah. You can get genus pretty reliably. Sometimes species is uh, I would say most of the species can be identified pretty well, but there's some that are going to be a little bit questionable. But then, as you mentioned, for some of these things, there's variance within a single species, and it's hard to really factor in that. There was a really great paper from Lindsay Kalin's lab, I think, that explored 2 different Staph aureus strains, and you could have vastly different outcomes based on those strain differences, and that's something that's really hard to factor into this, for sure.
Can you talk a little bit about the climate differences and how do you control for depending on when those samples were isolated, it could be hugely different from summertime to wintertime. And then hospitals also seem to spike their different wound pathogens. When you have cross-contamination, hospital-acquired infections, you may be, shifting the microbiome to one set of species or the other. Can you, very high level, could you give us insight on that?
Yeah. So as we were getting into this, so summarizing the weather data, so we took in, I want to say it was like 95% of all the samples that we had, we were able to associate each clinic to what's called an ASAWC, or a automated sensing. It's basically what we use to estimate the weather. And all of our clinics, or most of them, had one of these stations very close to it that I could extract daily summaries from, so I could have kind of this real-time picture of what these things like temperature and humidity and all those other things look like for each clinic and at the times that the samples were taken. So each measurement was, corresponding to like the day of collection. And then on top of that, and I think more to your point here, we are able to kind of factor in both proximity of samples as kind of a factor. So, if in this case we're using the clinic as the kind of the geolocation, and so if there's samples that were taken from the same clinic, that would register as being in very high proximity. And there were had to get, so for this part, I had to get with a spatial analysis expert from our NRM department. And we worked out a way of kind of accounting for that local effect. And there was a significant effect of, I believe the term is spatial heterogeneity, in terms of seeing that things that are closer together did actually, they were compositionally more similar. So there was that effect, and that was something that we could kind of work into our model and kind of account for, to make sure that our weather variables were not simply just accounting for that effect. They're actually accounting for what we would call unique variation.
So we all want to know if you have a chronic wound infection, and it depending on what time of year, what temperature and humidity, which hospital should I go to be treated?
But so I don't want to tell anyone to move.
Don't libel anyone now.
Yeah, I don't want to tell you.
No, but is there? So could you see? You talk about these complex biofilms, microbiomes, right? That have a lot of these anaerobic bacteria in a mixture, and they seem to have a very high bioburden, right? So was that persistent in the Midwest or in the South, or how did it kind of?
Yeah.
What did you learn?
Yeah, so there were a couple of extremes that stood out. There was an area, I can only think of calling it kind of like the Ozarks, this like central part of the United States, that was really, really high in one of the community state types that was dominated by anaerobes and whatnot. And then there were other ones that, say in around Arizona, where it's just really dry and hot, that actually seem to encourage more of those low bioburden types. So, there were definitely some areas that stood out more. I want to say the effect in general was a lot more pronounced at the extremes. And the other trend that we noticed was, so in addition to all the things I described, we also had quantitative PCR data on the detection of common resistance genes for all of these samples. And when we looked at that, aggregated across the different months, there was a very strong association between temperature and the detection of resistance genes. So as it's hotter, you do detect more resistance genes, and the effect was quite sharp. I mean, it accounted for a little more than a 10% difference in our frequencies of detecting.
I had a couple questions about that. I mean, for someone who lives in Arizona, that's not the best news to hear. But when you were generous enough to send us your paper, so I had a chance to mull this data over a bit. This beautiful plot that readers will see when they get the final paper, showing the distribution of the different CSTs across the US, and you point out that it's unevenly distributed, but there's these hot spots in the South, right? That happen to be also where the diabetes belt is in the US, right? Where you're going to end up having probably patients with more likely biofilm formation in their wounds, and they're going to be the ones going to clinics, and they're going to be the ones depositing. Although earlier on, of course, you point out that you get samples from everywhere in the country. So I'm wondering, to what degree do you just have stronger data from the South, where there tends to be more diabetes in southern Texas and Georgia and Florida, whether that is enriching for these sort of worse wounds that would have worse things like antibiotic resistance genes, and whether that can be extrapolated beyond, to what degree is the is the population driving the data result, or is the, weather and all that other stuff? You know what I mean?
Yeah. So it's definitely hard to separate those things because I mean we are limited by, this is what the data looks like, and it's very much
Yeah,
driven
yeah.
by, you're more likely to get these samples from certain parts of the US. Um, I like to think it's something we can extrapolate from because I think it does reflect real life though. I mean it's
Hmm.
if you overlay one of those maps with one of the United States Census like diabetes maps, the hotspots are the same.
Yeah, exactly. And I would expect that to be the case as well, yeah, I mean, I you get studies and you read it through and you're like, well, this just raises more questions even than I had before. Sometimes that frustrates you and sometimes it excites you. And with this paper, it
It's a little bit
excites
both.
me because I mean, it definitely shows that there's, some environmental drivers of these wounds. And that's, of course, important. We know from microbiome studies in the gut that even going from one institution to another will have dramatically different effects than, let's say, animal models,
Yeah.
right? And I experienced this personally in my postdoc, and in some cases, depending on the institution, one building from another building is a totally different gut microbiome. So it's not surprising, but it's beautiful to see that this is panning out in the real world in something like a human wound. One lingering question that I'm left with from the paper is, as researchers, wet bench researchers, we're limited by models in some respect, right?
Yeah.
A lot of what we do has to come back to a model at some point. And so for many years, people were just infecting wounds with Staph aureus and saying, this is a biofilm, this is an infected wound, and that's what it is. And clearly, we all knew that that was not right. It was a richer, more diverse community in the wound. And so over the last couple of years, people have been moving towards, well, you have to have Staph and Pseudomonas, and then that constitutes a polymicrobial biofilm. And then now people are taking it a step further. Mitch and his team are experts on these 3D printed biofilms that you can place on wounds and create these rich communities. So my question to you is, now that you've seen the truth in the US population, at least, how do we as researchers bridge that gap between a reductionist system in the lab and the reality of these ultra rich communities in the real world? And how do we recapitulate these effects?
That's a great question, and one that we've talked about with one of our collaborators quite a bit. And I think I'm going to upset you with one part of this, which is I'm glad you mentioned, kind of the advancement towards models with Staph and Pseudomonas together, because what we've seen is you actually encounter those two in a wound together much less than you might expect. They don't naturally co-occur.
Yeah.
So when you study them in a lab, you're kind of like forcing or creating a situation that doesn't seem to be bearing, at least in the samples that we have, it doesn't actually seem to be a scenario that's showing up, quote, in the wild very much. So I think part of my answer here is going to be really just, I think it allows us to maybe ask questions and maybe reshape our models to fit this new glimpse of real world data. So what one of my collaborators is trying to do right now, and this is kind of I'm sure you can appreciate growing different bugs together can be a challenge. But one of the things that we're kind of moving towards is also developing new kind of model combinations to kind of recreate some of the community state types that we've seen. So maybe Staph aureus and Pseudomonas don't occur together that much, but what does Staph aureus occur with, or what does Pseudomonas occur with?
Yeah.
And reshape and rebuild your models from those to have a new kind of community that reflects that these different community state types that we're seeing.
Yeah, I think that's the natural next step, and honestly, it's going to be a paradigm shift, I think. I'm at the career stage where I'm sitting on study sections, and I'm like, oh, well, this is what it takes to get funded, and then I see the paper come out the next month, like, oh, well, everything that the study section said it takes to get funded is actually not true. So what are we actually doing? And we can only know what we know, and work like this that really highlights how far off we sometimes can be, I think, are of tremendous value to researchers. So
I just want to
I-
mention my former background. I did a lot of food safety inspection service stuff. So I've covered all the food pathogens and all the outbreaks in the country. And it was amazing to me every year the spikes of pathogenic E. coli in the summer and when a Listeria monocytogenes shows up and so on. And I love these kind of studies that really bring home the truth that we're studying complex polymicrobial environments, and that you're absolutely right that Pseudomonas doesn't always play nice with Staph aureus, and there's other combinations that really seem to be very robust biofilms. Sometimes we see when you have these two fighting together, they actually Staph may take over early, and then Pseudomonas will come in and get rid of the Staph. It's pretty amazing. Anyway, thank you kindly for this wonderful session for the WHS. Jordan, thank you also for being a great host, and look forward to posting this up on the website in the next week or so. And are you going to be at SAWC again this year, WHS?
I'm planning on it. Yes.
Where's the meeting this year? Is it Texas? Is it?
Yeah, it's in Texas. Yep.
Okay, that'll be fun. No. So
Well, I don't know. There's a lot of CSTs in that area, so I don't know.
No, exactly. So next time you get a chronic wound, come to Massachusetts where there's less issues. No, I'm just kidding. Anyway, take care. Great to see everyone. Have a wonderful
Good to
day.
see you guys. Happy New Year.